The G protein functions as a molecular switch that either turns on or off depending on whether a GDP or GTP is attached. When the appropriate signaling molecule binds to the extracellular side of the receptor is activated and changes shape. The cytoplasmic side then binds to an inactive G protein, causing a GTP to displace the GDP, activating the G protein. The activated G protein then disassociates from the receptor, diffusing along the membrane, which then binds to an enzyme. Once the enzyme is activated, it can trigger the next step, leading to cellular response. The G protein then leaves the enzyme, which returns to its original state, which is then available for reuse.
This passage describes a ligand-gated ion channel receptor, which remains closed until a ligand binds to it. When the ligand binds to the receptor, the channel opens, allowing specific ions to pass through the plasma membrane. This ion flow rapidly alters the concentration of those ions inside of the cell. For example, ions like Na plus and Ca2 plus can diffuse into the cell when the ligand binds to the extracellular domain of the receptor. Once the ligand disassociates, the channel closes and the ion flow stops. This process is crucial for neuronal signaling and other cellular functions. Additionally, ligand-gated channels can interact with other receptors such as GPCRs or G protein-coupled receptors.
This diagram shows the signaling process of a receptor tyrosine kinase pathway, specifically through the EGF receptor. RTKs are cell surface receptors that, when activated, trigger signaling in the cell which cascades promoting cellular growth and division. When EGF binds to its receptor, the receptor goes through the pairing and autophosphorylation and then activates the intercellular domain. RAF phosphorylates and activates MEK. Activated ERK phosphorylates cytosolic targets and transfers into the nucleus, where it phosphorylates factors such as C-MYC. These factors make it possible for the expression of genes involved in cell growth and division. The pathway is regulated and crucial for normal cell function, but can lead to diseases like cancer if it's undone. Lastly, mutations may result in uncontrolled growth signaling.
Cells use intracellular signaling to respond to signals from the environment and other cells. An example of intracellular signaling is with the hormone aldosterone. Aldosterone first diffuses through the membrane and binds to its receptor. Then the hormone receptor complex diffuses into the nucleus and binds to the DNA. After this process occurs, transcription and translation take place. After transcription and translation, a cellular response is produced.